ideas. What you consume influences who you become. On the Mind and Matter podcast, we learn together from the best scientists and thinkers alive today about how your mind-body reacts to what you feed it. Before starting Mind and Matter, I spent 10 years in academia doing scientific research. I got a PhD in neuroscience where I focused on neuroendocrinology and the neurobiology of behavior, and before that, I specialized in molecular, developmental, and evolutionary genetics. I use my scientific background to help parse and translate the information that guests share on the podcast. In addition to the podcast, I write long-form written content inspired by the show where I integrate what I've learned across episodes. I also have a free weekly newsletter where I provide you with upcoming guests, share links, and provide commentary on scientific studies and research that I'm reading, and more. Visit mindandmatter.substack.com to find all of my content. You are about to listen to the full episode, but there will be 1 to 2 short ads immediately following this before the conversation. If you would like to hear ad-free episodes and get early access before they release more widely, become a paid subscriber at the Mind and Matter Substack at mindandmatter.substack.com. Becoming a paid subscriber is a way to directly support my efforts, allowing me to make more episodes and higher quality episodes, and also helping to free up my time so that I can spend more of it on doing research and synthesizing results from the podcast and from the literature so that I can do more things like writing on the Substack, putting more content into the newsletter, and getting these things out more frequently. So if you want to support the show and get ad-free episodes, go to mindandmatter.substack.com. Hey everyone, I want to take just a minute to tell you about a really cool health monitoring device I've been using called Lumen. It's a handheld, pocket-sized device with a sleek design. The Lumen device measures CO2 levels in your breath, which allows their technology to determine the extent to which your body is burning fats versus carbohydrates. Lumen helps improve your metabolic flexibility, your body's efficiency in shifting between using fats and carbs for energy. I use this device in the morning or before bed or after meals or after workouts to track my metabolism. With just a couple weeks of use, I learned a lot about which foods were causing my body to burn mostly fat, mostly carbs, or both, as well as how long I needed to fast and how hard I needed to exercise to promote fat burning. Being able to track whether your body is burning fat versus carbs will help you figure out how your body responds to your lifestyle and whether or not your weight loss and fitness strategies are actually working. Lumen is great for anyone looking to optimize their health for either weight loss or athletic performance. Their easy-to-use app allows you to track your results together with what you're eating and how you're exercising, and it syncs with other devices like the Apple Watch. Click the link in the episode description to learn more and use the code MINDMATTER, MINDMATTER, to get 10% off your Lumen device today. That's on top of the site-wide sale they are currently running, and the Lumen device is eligible to be paid for with FSA and HSA health accounts. Check out the link in the episode description for more info. Justin Perry, thank you for joining me.
Yeah, thanks for having me.
Do you want to just start off by telling everyone a little bit about who you are and what your lab studies?
Yeah, sure. I'm Justin Perry. I'm first and foremost a card-carrying Alaskan, but I am by training an immunologist and also a clinical psychologist. So that's kind of what informed a lot of our recent work. And what we'll be talking about today is what my lab really focuses on, which is the cell clearance machinery that our bodies have to deal with dying cells, dead cells, debris, et cetera, and how those maintain homeostasis and how those go awry in disease development and so on.
Yeah, so there's a lot of stuff in our bodies all of the time that needs to be cleaned up, like literally, for lack of a better term, trash, gunk, broken things, debris. Can you just talk a little bit about that on a very high level? Like what is generally like the stuff that gets cleaned up when you're not sick, just you're living your life day to day, waking up, going to bed? What is the stuff that gets cleaned up?
Yeah, so we have one of our favorite lines whenever we give a talk, either to scientists or to the public, is that the human body, the average human body, which is, usually the white male, 25 to 45, is composed of about 30 trillion cells, and about 1 to 2% of that biomass, those cells turn over every day. So if you do a little back of the napkin calculations, that's 3 million cells per second. So that's a major form of stuff that's turning over. The vast majority of that is red blood cells, neutrophils. About 90% of it is that.
So most of it is cells that divide and reproduce very quickly. There's a lot of turnover.
Yeah, there's a lot of and they do their jobs pretty quickly and then they're replaced pretty quickly.
Hmm.
So what I was going to add is though that, if you take a step back and actually look, there's two periods, and we'll talk a little bit about one of those today, which is it turns out that in children and in women, that rate is double. So in women, the number one tissue that turns over is the endometrium. It's actually
Mm.
really poorly studied. We don't know a lot. And then in children, it's neurons, dying neurons, these sorts of things, right? So all of the unused tissues and cells as you're developing have to be eliminated. And so in children and women, it's double that.
Mhm. So you mean women generally speaking, not pregnant women, just sort of the monthly turnover that happens with the female body.
Yeah, exactly. Exactly.
Wow. Wow. So double the rate of cell death and stuff that needs to be recycled, cleaned up, and replaced.
Yeah. And then we're not even really including all of the other biomass. So in your lungs, the way we breathe is through gas exchange. That gas exchange requires a substance called surfactant that has to coat in the alveoli and help facilitate that process. And so in your lungs, you have cells that do this phagocytic clearance to help turn over that surfactant. And if you don't do that, you get diseases like cystic fibrosis, et cetera. So there are specific materials and substances in every tissue as well that help, or that your phagocytes, such as the cells we study, macrophages, that facilitate that process.
And then, using whatever example you want to illustrate this, can you just give us a sense for how this starts to work at the cellular level? So if we follow a red blood cell or something like that, it lives, it does its thing, and then it dies. What is the cleanup process like at a basic level?
Sure, yeah. So it's we characterize this in basically four steps. So we like to simplify things in terms of eating analogies. So basically, we have a series of signals. So when a cell dies, it has to tell neighboring cells that it just died. We call that a find me signal. So you'll hear this occasionally, these signals called find me signals. Those are molecular signals like ATP, some other metabolites that are released that basically tell the neighborhood that I've died. I'm a dead cell. However, when those clearing cells, we'll just call them phagocytes. When that phagocyte comes to that area, it has to distinguish death and life. And that's really important, right? You don't want your phagocytes eating your live cells.
Right, right.
And so the way it distinguishes this is through another series of molecular patterns. One of them is a phospholipid called phosphatidylserine. And that's interesting because phosphatidylserine is really abundant inside of the cell. And then when a cell dies, like a red blood cell or neutrophil, it flips that signal out. That signal has to be recognized. That's the main way that we can recognize life and death. That shouldn't be there unless that cell is dying.
Right.
And then when that recognition occurs, that triggers an internalization event. We call that, those are the eat me signals that initiate the engulfment of those dead cells, dead or dying cells. So that's the third step. And then the fourth step, which is what my lab specializes in, and we're really pioneering the study of, is what we call digestion signals. So literally, when one cell is eaten another cell, how the heck do you deal with all that biomass, right? All of that stuff that you've just eaten. There's got to be a lot of, signaling and dealing with the material. And so those are digestion signals. So that's really sort of the arc of that process.
So a cell dies and basically its body is ruptured or broken somehow, such that stuff that's normally fully contained on the inside starts leaking out. There's ATP and stuff floating around. It's almost like an animal that smells a dead carcass that attracts these phagocytes. And then they are reading the molecular signals around the area, and they're looking for stuff that means dead, which is essentially stuff that's normally on the inside of the cell is now exposed and it's on the outside, and that's the signal for eat me.
Yeah. Yeah. And it's actually regulated. So this is very important because essentially you have what are non-inflammatory. So things that don't cause inflammation is what this normal regulated cell death process called apoptosis, right? And in fact, that's non-ruptured. So it's very controlled. And in fact, I like to say that when a cell dies, it forms a self-contained, ball of material. So, in fact, in inflammatory forms of death, which we can talk about, So those are one of famous one is called pyroptosis. It's a very inflammatory, often caused by infection.
Hmm.
That's when that stuff starts leaking out. In fact, you
Mm,
don't want that stuff to be leaking out
right,
because that can trigger,
right,
an
right.
immune response.
So cells can die on purpose. That's programmed. It's baked into the cell
Yeah.
itself. It's highly regulated. And then cells can die on accident, which can come with extra, that can come with side effects or potential problems that come from something puncturing the cell or breaking it open that then, might cause inflammation and,
Yeah.
and other stuff.
Yep. And if you block the regular one, the regulated one, and you don't clear that dead cell, this is sort of the classic way of thinking. Maybe that would result in autoimmunity like lupus.
Mm,
Lupus is a classic disease because then if you don't clear it, you start leaking those things out. Like you leak DNA, you leak,
right.
right? And then that leads that classic hallmark anti-double-stranded DNA. So you develop antibodies against that. So that's one example of where failure to clear those dead cells at all
Mhm.
leads to what's called necrosis, and then you have sort of an immune reaction to that.
Yeah, yeah, wow. Yeah, there's lots of potential analogies we can draw here. So
Mm.
this process can go wrong, and I definitely want to talk about that. But let's talk about phagocytosis when it goes right.
Yeah.
One of the things I want to talk about, so let's just define phagocyte for people. Is that just any cell that can engulf and eat other cells? What's the specific
Yeah.
definition?
And we love this terminology because so we have professional, non-professional. We're scientists. We like to characterize everything, right? So it's professional, non-professional, and specialized. But their phagocytes are literally anything that can, use its own ATP to internalize another cell.
Mhm,
And phagocytosis isn't just other dead cells; it can also eat bacteria.
mhm,
It's just that process of literally eating stuff. We study, the immune cell called a macrophage, big eater, literally macrophage. But other cells can do this as well. And the classic example I love to give is um, post lactation. So pregnant women, after they've had a baby, they lactate. And after lactation is done, there's a massive involution process. So you have to, half of the mammary glands that have expanded out have to,
mm,
they undergo death. You have to clear all of that material. And a lot of that is done by neighboring epithelial cells. So the
hmm.
neighboring cells in those mammaries can actually do this themselves. That's a non-professional role or non-professional
Because
phagocyte.
it's not their main role. It's not what they're
That's their
specialized
main job.
for, but they
Yep.
can do it in the right context.
They can do it right. Exactly. And then the third one is specialized. And that's a classic example of this is in your eye. So each morning we wake up, we open our eye, light hits it. This is an important part of color vision. But as part of that, you produce reactive oxygen in the eye.
Mhm.
And so in order to prevent damage in the rods, they literally shed their outer segments, and those are eaten by neighboring specialized phagocytes called retinal pigmented epithelial cells. So that is their job, but they're not a macrophage.
Mhm,
They
mhm,
just do that specific role. And another good example of that would be the Sertoli cell in the testes, right?
mhm.
So we turn over sperm, billions of sperm. Those have to be cleared, and those are cleared by a specialized phagocyte called Sertoli cell.
Hmm. Yeah, yeah. So it's like there's literally a garbage man, and it's his job every day to clean up the garbage. But like other people know how to take out the trash. It's just not what they do professionally.
I love this analogy because I've joked for a long time if I could draw, I would do a children's book where I would have the body be a neighborhood. Each organ and tissue is a different neighborhood. And, then you could characterize the roles that you have the garbage collector, you have the detective, you have, and so these macrophages or different phagocytes are all serving these roles in that neighborhood.
Mhm.
Everyone's essential. They all do an essential thing. But one of them really is this, as you say, the garbage collector.
Yeah. So staying on the subtopic of phagocytosis when it goes right and everything's working properly, I want to ask you about, so phagocytes are engulfing other cells. They can be apoptotic cells that have been programmed to die and they were supposed to do that. They can be cells that got sick or poisoned in some way, shape, or form, and things have gone wrong, and then we need to get rid of those. They can be foreign cells, like bacteria cells. In many of these cases, the cells will contain something nasty. Maybe it's literally a poison. Maybe we're actually talking about a bacteria. There's lipopolysaccharide in there. There's stuff that can cause problems for the body. When a phagocyte engulfs such a cell, does it have specific cellular adaptations that prevent it from getting sick? How does it deal with the toxins and stuff like that?
Well, and I want to be really clear too. I mean, this is an exciting area of the work right now. So I don't want to pretend like we have all the answers. This is literally the I think our lab and many labs are trying to figure out, because it turns out, for instance, uh, there are bacteria that can induce apoptosis, not just like bad cell death,
Mm-hmm.
but normal homeostatic cell death. And so your macrophages, you can imagine, I hate to anthropomorphize too much, but evolution wouldn't have selected for, these cells to assume there's no infection present. They're going to, presumably be primed and ready to deal with an infection, right? Like think about it like the homicide detective analogy. So, you don't want to assume that this is a suicide. You always look for clues and
Right,
signs and
right.
evidence of a
Yeah,
homicide.
they're going to try and figure things out. They're not just mindlessly taking
Yes.
out stuff.
Exactly. So we do know of some classic sensors, highly evolved and evolutionarily conserved. The toll-like receptors are a
Hmm.
classic example. We have certain toll-like receptors that are in the phagolysosome that can sense this. However, many of those are reactive to self molecules like mitochondrial DNA. So that's why your question is so fascinating, because you have to have systems in place that don't respond to those potentially dangerous self molecules like mitochondrial DNA, like modified amino acids and things that, you're breaking down that biomass, and a lot of that it's reactive, dangerous
Hmm.
material.
Hmm.
And so you have to be able to deal with that. And so, I think again, like I said, this is an emerging area of work, but the that internalization process normally doesn't cause what's called lysosome stress, right? So the
Hmm.
lysosome is an organelle that's essential for breaking down the stuff you take up.
Hmm.
And normally your lysosomes have all of the enzymes and the capacity to break that down,
Hmm.
and that's not stressful. But if you have a pathogen in there, one example could be that you change the cell volume of the lysosome or you
Hmm.
change the acidity, et cetera. And that can be sensed too, by different
Mm.
protein sensors, in your phagocytes. And so that's actually really exciting. And there was recent work looking at like glyco modified RNAs as an example, right? So just, so these like, um, sugar modified RNAs might be sensed, and so we're really trying to figure that out. But yeah, you're completely right. That
Hmm.
is really probably the way that this is sensed is, not quite self or not quite right is
Right,
what our
right.
phagocytes are sensing.
And in terms of the capabilities that say a macrophage has, so this is the garbage man. This is an immune cell that's supposed to eat things. That's its entire job. I would imagine that it's got some special characteristics or some extra firepower in certain capacities. Maybe it's got more lysosomes or something. I'll let you tell me that compared to other cells. So, just to use our analogies here, like if I was the garbage man or I'm handling chemical waste as my job or whatever, right? I'm going to have gloves and protective material. I'm going to be exposed to more nasty stuff with pathogens. I'm going to have stuff that's part of my job that helps protect me from being exposed to all those things all the time because that's what I'm doing. What do macrophages have that allows them to eat eat
Yeah.
debris and eat pathogens and stuff like that?
Yeah. So, I mean, you hit the nail on the head. I mean, the first one really is are all of these what we call innate immune sensors, right? These proteins that are capable of sensing, um, patterns,
Hmm.
recognizing different patterns that aren't supposed to be there,
Yeah.
like motifs that aren't supposed to be there. That's the classic view. Um, the other one I would add is really, you know, very early on during development, our macrophages seed tissues and are educated by the organ and tissue that they emerge in.
Hmm.
So if you have a macrophage that goes to the brain, we'll talk about this. They're called microglia, and they are really developing with that tissue, and they're being informed
Hmm.
by the oxygen, the metabolites,
Hmm.
all of the different stuff that makes up that tissue, the processes that are going on. And that also helps educate them transcriptionally and gives them the tools to sort of facilitate and support that tissue.
Hmm.
So I think that's something that's a really exciting area where, more evidence is emerging that it's not just these, all macrophages have these innate immune sensors,
Hmm.
but there's also specialization as well, right? So those macrophages are capable of rapidly transcribing and translating new proteins in order to adapt and sort of exist in those
Hmm.
tissue environments. Often, for instance, for your microglia, your entire life, right?
Yeah.
So
Yeah.
most of the microglia you're born with, you have your whole life.
Yeah. So different tissues obviously have different properties. Like the brain is very different from the liver, is very different from the stomach or whatever. They're going to be exposed to different levels of reactive oxygen species. Different cells are in there. Different pathogens can get in there. And so you're saying that macrophages can become educated. It's almost like they go to school, right? And one majors in this, one majors in that. They sort of learn how to specifically work well in a particular tissue. At least some of them do.
Yeah, and I'll give an alternative example, which is a macrophage called an osteoclast. So the osteoclast is the bone resident macrophage,
Hmm.
and those macrophages become educated on bone, basically. They're educated and optimized to recycle and remodel your bone, right? So if you don't have them, you can't remodel bone, you develop osteopetrosis. And in fact, probably one of the major issues with aging is their decreasing capacity and function to remodel bone as we get older, sadly, right? Where I know certainly I'm more and more as I go is my bone remodeling is probably doing less and less quality activity. So that's another example where that macrophage seeded early on during embryogenesis, during the development periods, and um, gains this capacity through environmental cues signaling to it in order to optimally facilitate bone remodeling.
Hmm. Hmm. And part of where we're going to go with some of this is that when this process goes wrong, it's obviously going to lead to problems, right? Just like if you don't take out the trash in your home, if you let clutter and stuff build up, there's going to be fire hazards. Stuff's going to become rotten and rancid. Like stuff can very quickly go wrong. There are certain times in life where this is especially important, and you've already hinted at this. Women have a lot of turnover as part of their monthly cycles. I would imagine pregnant women. I mean, you mentioned the mammary gland example, right? She has to build new tissue to support the baby growing and then get rid of it after things are done. And then young children when they're developing. In particular, the human brain has to do a lot of synaptic pruning early in life. There's sort of an over exuberance of stuff built and then it's pruned away. And I would imagine that the pruning away part, the taking out the trash part is critical. And when that goes wrong, it can cause a lot of problems.
Yeah, so I think we'll take this in two parts because I think the sort of the conventional classic way of viewing this that for 50 years since we've defined even how cells die, we've kind of known that they had to be cleared. This is called what I would call the failure to appropriately clear model of disease development is exactly what you said. If you don't clear that material away, then it can arise and it'll lead to issues. And so I think probably because of what you said, and let me give an even more striking example. There's a pretty famous study from one of our pioneers, a guy named Shige Nagata. They showed that actually, and this is why I say the macrophage is the most important cell, much to my other colleagues' chagrin, is it turns out that in during development, your red blood cells, so your early phase of red blood cell development, they have to enucleate. They get rid of their nucleus. That has to be cleared by embryonic macrophages.
Hmm.
And if you don't do that, you die during embryogenesis.
Hmm.
So without macrophages clearing those nuclei, the nuclei from the enucleated red blood cells, you know, you don't develop. And so that's like a classic example of really needing to take out the garbage. And so it's such a highly evolutionarily conserved process that we have redundancy in receptors, we
Right,
have
right.
redundancy in proteins and molecules. This is probably why we don't see a lot of disease arising because of this failure to clear at all,
Hmm.
right?
Hmm.
So
It's so
we
harmful that the embryo can't even form. Like it's
can't
it's
even
it's
form.
really
Yeah,
baked in.
yeah, exactly. And so there's our host of receptors. I talked about the find me signal. There are receptors, sorry, the eat me signals. There are a host of receptors that recognize those. And if you try and genetically delete one in a mouse or a rat, there's another five that can compensate
Right.
for it,
Right.
right?
Yeah, and that's a
And
general principle I would say in biology, right? That when something is like truly, truly critical, there's often redundancy built in so that you've got backup systems in place because everything would fail in the absence
yep,
of this ability.
absolutely. And it's also often why those systems are co-opted by pathogens. So just as a fun, interesting aside, many of the receptors we know viruses use to get in are these recognition receptors.
Hmm.
So the viruses actually exploit this conservation to
Yeah.
their
Yeah.
advantage.
There's always that. Yeah. That lock is always going to be there to be picked because we need it to
Yes,
be a certain way.
we need it. And so then the other way of viewing sort of that this, disease development, where it goes awry is what my lab is specialized in, is what we call that digestion process. And so it turns out that even the hallmark autoimmune disease that we associate with the failure to clear dead cells is lupus. It turns out the original
Mhm.
diagnostic criteria for that,
Sorry,
yeah,
can
yeah,
you just,
please.
what is lupus exactly? Because I'm not even sure I know.
Yeah. Well, okay. Well, it's without offending any clinicians, I mean it's basically a multi-organ autoimmune disease where you form an immune response, T and B cell response against autoantigens.
Mhm,
So
mhm.
the classic being double-stranded DNA,
Got
right?
it.
So you have antibodies. Those antibodies deposit in tissues like your kidneys or multiple organs and
Yeah.
tissues,
Yeah.
and then your immune cells attack it, right,
I
and
see.
kill
So it's
it.
autoimmunity at a fundamental level because double-stranded DNA is in a lot of places, and so it's a
Yeah,
multi-organ autoimmune issue.
exactly right. And that's what makes it so difficult to come up with like a non-umbrella diagnosis
Right.
of
Right.
this because patients will present differently depending on a host of different factors.
Right. My kidney flares up a little bit more than your other
Yeah.
tissue, and but it's still the same
Yep.
fundamental thing.
Whereas perhaps my thyroid gland will be attacked, and then I'll have psychological symptoms and hormonal symptoms, et
Mhm,
cetera.
mhm.
So yeah, it's
So before getting into some of your newer research and talking about microglia specifically, although we could use them as an example, can you
yeah.
walk us through? So let's say a macrophage engulfs an apoptotic cell or something. You can pick whatever example you want. What does that cellular digestion process look like? When a macrophage engulfs another cell, what are the main steps inside the macrophage that are breaking down that cellular stuff?
Yeah, so we have a pretty decent understanding of at least the basic machinery of how this works. And so after pick an apoptotic cell is internalized by a phagocyte, you then begin a slow sort of, acidification process, right? So you have to begin to acidify that because a lot of those breakdown enzymes depend on very specific pHs.
Right, right.
Um, and so, in order to achieve that, your macrophages or your phagocytes start recruiting lysosomes, which are your organelles that contain a lot of those pH sensitive enzymes.
Hmm.
So you
Is
have
it fair to say that the lysosome is almost like the stomach of the macrophage or the cell, and we're going to send stuff into this acidic bag to be broken down?
it's almost like the things that make the stomach acidic, and collectively between the what we call a phagosome and the lysosome fusing together would make your stomach, right?
Hmm.
And so the lysosomes are literally just the things that like the gastric juices
Hmm.
essentially. It's giving you the gastric juice and the enzymes. But there's an important aspect of this that often is forgotten. Once you fuse lysosomes, multiple lysosomes to a phagosome, let's say you've successfully broken down all of the material. You've recycled your DNA and RNA and amino acids and lipids and right, all of the stuff you have to do. You then have to resolve that that whole stomach, right? Like unfortunately, you can't just leave that in place because it is a dangerous organelle. And so then the final step is literally the breakdown of the what we call the phagolysosome, right? The fused formation of those things. Then you have to recycle that material as well. And so our cells then have systems in place to, it's called tubulation, literally the process of pulling the lipids away and recycling them in the endoplasmic reticulum. And so basically you build this thing and then you break it down. And then you do it again. You build this thing and then you break it
Hmm.
down. And what
Hmm.
makes a professional phagocyte so good is that they can do this much quicker than any other cell
I
can.
see. So they've got components and machinery that are just really fine tuned for quickly and efficiently doing this type of internal digestive thing.
Yes, yeah, absolutely. And that would be probably the one big difference I would say between something that is a professional versus not.
Mm-hmm. And are there any special metabolic capabilities or needs or components that need to be hooked up in something like a macrophage to enable all this stuff to happen? Are they generating ATP or utilizing substrates like glucose or fatty acids differently than most other cells in order to power all of this specialization?
Yeah, so this is talking about expertise. This is one area where my lab focuses a lot on is to try and understand, not just the energy requirements, because the thing you always have to remember is that this isn't happening in isolation. We love doing cell culture work, but in reality, these cells are operating in a network of other cells. And those other cells, like in the brain, your neurons, et cetera, they need that glucose. They need those other molecules and that energy to do their own stuff. And so, macrophages particularly are fine-tuned to use not just use substrates like glucose, but use them efficiently. And what I mean by that is that they have to be able to use whatever's in the environment to both fuel that ATP, like you said, to give, generate enough energy to make that phagosome and to eat that dead cell, but then also efficiently because they consume a lot less of those substrates of things like glucose and use them in unique ways in order to acidify the lysosome, recycle the biomass, et cetera. And so while we don't have a full picture of this yet, that is something that we're learning is really, really remarkable ability to sort of adapt to what are really harsh environments, right? Often
Yeah.
low oxygen availability, low substrate availability, and
Yeah.
yeah,
Yeah, it's almost like, I mean, we use the garbage man analogy, but I'm almost thinking of a firefighter here. They have to go into these hostile environments. They don't know what the oxygen or the air is going to be like, and they have to be able to deal with things probably in a very flexible way because, I mean, even if I bump my knee or I get a run of the mill infection, you probably can't know if you're the macrophage ahead of time, like what's the glucose concentration they're going to be, like how tightly are things packed. You just have to be able to kind of use whatever is in the environment locally.
and just think of the classic example we teach in class where you get a paper cut. So you get a paper cut, you've now opened a wound, and a macrophage, or in this case, is not only having to sort of assess the damage of the cut, the influx of potential pathogens, but they also have to support the wound healing process as well, right?
Mm.
So you have fibroblasts that need to make collagen, you have epithelial cells that need to proliferate. So those macrophages are not only dealing with the initial damage, they're also assessing what's going on and sort of fueling, if you will, these other, essential reparative processes, right? And so, I think we'll get into the cancer side, but that's literally what cancer exploits is a macrophage's desire and need to support that sort of wound healing proliferative response.
Yeah, yeah. Are there any so similar question. Are there any special cellular features that macrophages have that allow them to use energy sources that other cells can't use or to take up extra glucose, fructose, whatever from the environment, like anything like that, where like they can just tap into fuel sources in a way that a normal cell cannot?
Yeah, so I'll give you an example from something we published. But the broad answer is absolutely. As part of this adaptation, they upregulate and differentially regulate enzymes that allow them to do these different metabolic processes. So the example that we published on is in these sorts of low oxygen environments that most of your macrophages live in, right? So most tissue oxygen is actually quite low. And macrophages exist in the deep tissue, what we call the parenchyma of the tissue. They have, they're far from the vasculature, so they're not getting a ton of glucose, and they have really low oxygen availability. And so they adapt to these settings by basically what little glucose they can take, they shunt it into an alternative metabolic pathway called the non-canonical pentose phosphate pathway. So literally that pathway normally is used to make two things: nucleotides, biomass,
Hmm.
and what NADPH, right? And NADPH is an important redox metabolite. And so our macrophages are exquisitely tuned to be able to shunt what little glucose they can take up into this pathway that basically cyclically generates NADPH to control redox stress, but also use that NADPH for lysosome acidification, another thing that's important for.
Hmm. Hmm. Got it. So it's almost like because they have to spend so much of their lives in the parenchyma away from the blood vessel. So if people imagine a blood vessel, it's delivering all of the stuff to cells, but then it's got to diffuse some distance. And the further you are from the vessel, right, the longer it takes and the less concentrated the nutrients, the oxygen, all that will be. So if you're like a little cell living right by the supply, it's almost like you were born into a wealthy family and like you just
Mm.
had everything you needed. But if you're growing up in sort of the rougher neighborhood, you've got to figure out how to use things more economically.
Or as an Alaskan, if you grew up in rural Alaska like I did, right? That's the other one is you got to be able to hunt and forage and
Right.
salvage for your own
Right.
stuff. Yeah.
Interesting. Okay, so they've got lots of adaptations that
Yeah.
enable them to efficiently do all this digestive stuff, and that requires we'll just call it the creative and economical use of energy. So they are able to engage in do things with energy metabolism that are a bit different than other cells. Let's talk about glucose and fructose next, because we're going to talk about this fructose story coming up. So glucose is just sort of the probably the most famous molecule for how we generate energy in our cells. I don't think we need to go into the basics of mitochondrial metabolism in this episode. We'll just assume people know something about that. Glucose can be used to create ATP. How is fructose different from glucose in terms of how most cells either do or do not utilize it?
Yeah. It's actually coming into this area really striking because, when we eat, putting aside high fructose corn syrup, which of course has been an important nutrient additive and good or bad depending on who you talk to, right? Historically, most of us get our glucose from sucrose, right? So sucrose broke down into two molecules, glucose and fructose.
Mm-hmm.
And so our bodies then really heavily rely on the glucose. Fructose, on the other hand, is not really, well, historically has been thought of as kind of just a byproduct of that, and it's not really used, right? So um, you consume sucrose, it's broken down, that fructose has to be recycled, so it's absorbed by the small intestine, shunted through the liver, but then a lot of it is just excreted out, right? So what little you use for lipid storage in the liver, the rest of it is just excreted out. That's the classic way of thinking of it. It's not a primary um, source of energy, and that's because the vast majority of the cells in your body either don't have the ability to take it up, what we call transport, or they don't have the appropriate enzyme to phosphorylate that fructose,
Mm-hmm.
right?
Mm-hmm.
Or it's just that those cells are so dependent and reliant on phosphorylating glucose that the idea that they would also take up fructose is probably negligible. So I mean, and probably alluding to past guests too, one of the main things that fructose is converted to is lipids. And so
Mm-hmm.
that is when you consume too much fructose, that is a case where you can then convert it into lipid storage. And those lipids can be used. It's obviously harder to break those down, but you can use those for energy use. And then the other way is what we call the polyol pathway. And this occurs in your liver, where you're literally converting, it's kind of like gluconeogenesis, but it's gluconeogenesis from fructose. So
Hmm.
you're basically making glucose from fructose via this polyol pathway through what's called sorbitol intermediate, right?
Mm-hmm. Mm-hmm.
And those two are interconvertible, glucose to fructose and back again. And
Mm-hmm.
so a lot of people who, even in the brain, people have observed, they thought if any fructose is even getting to the brain, it's probably just converted to glucose.
Mm-hmm. Mm-hmm. I see. So there are situations in which, although fructose is not used itself as a fuel source in many cells most of the time, fructose can be used in certain contexts to create more glucose.
Yeah, yeah.
So what about macrophages? Are they able to suck up fructose from the environment? Do they have those transporters?
Yeah, so we'll get onto this, but I mean, one of the first things I always like to do when I come across something interesting, an interesting protein, is just look at, data sets that have basically sequencing of every macrophage. And it turned out that if you look at the primary transporter for fructose, GLUT5 or SLC2A5, it's not expressed in any macrophage in humans or mice or rats or monkeys other than one. And that's the brain macrophage called microglia. And
So
so
there's
that was
many.
really like, yeah, go ahead.
Yeah. So as we said earlier, there's many different types of macrophages. Many of them are specialized for certain tissues. You're saying that none of the macrophages have this transporter that allows you to grab fructose from the environment and internalize it, except the macrophages that live in the nervous system.
Yeah. Yep.
And so let's talk about microglia for a minute. So these are the macrophages that live in the nervous system. What? I mean, there's a lot we could go into here. I don't want to go too deep into the weeds, but can you give us just a basic, simplified sense for how were people thinking about microglia up until not that long ago, and what have we started to learn recently about the totality of what they're doing in the brain?
Yeah. So, you're alluding to a really interesting point, which was up until I think many groups would credit the the Barra's lab in California. Up until that point, the neuroscientists generally thought microglia weren't really that important of a cell. They were just a small glia cell, and it wasn't, they probably did something, but it wasn't super clear what they did. But the main focus was, you have astrocytes that are dealing with a lot of the toxic substances. You have oligodendrocytes that are important for myelinating and remyelinating, et cetera. And then you have neurons that are the money, right? That's really where the action is in the brain. Until the Barr's lab and many of the people that been trained came across this, observation that actually it turns out that they are a macrophage, and B, they are phagocytic, and they're doing important things. First, it was really clear that they were doing it in the context of demyelination and regeneration and remyelination. Then it was observed that they're probably important either preventers or contributors. We're still figuring that out for a neurodegenerative disease like Alzheimer's, et cetera. And then it was like, okay, really looking at the development question, right? This early windows where we already knew that you made way more neurons than you are going to use in your lifetime. And so you have to prune those back, as you said, what we call synaptic pruning. And also just generally, half of your neurons die, and those have to be cleared by something. And that's, more, last decade or so, data's accumulated that those microglia are important for the phagocytic clearance of dying neurons.
Yeah. And to make it clear for people, if it's not already, like when we say synaptic pruning and cell death in early life for humans and other animals, like a lot. It's a massive amount. It's not just like we lose a few synapses. It's an incredible amount of connections and cells that have to go away. And if that doesn't happen, your brain won't work properly.
Yeah, to go back to my math that has been actually tabulated by groups much smarter than me, doing these sorts of physiological calculations. I told you it's 3 million cells per second. If it's double in children, and the vast majority of that is in the brain, that means about 2 to 3 million cells per second in the brain are going away. And every one of those neurons, there are some unused synaptic connections. So it's at least one per neuron, perhaps more per neuron, right? That also have to be, as you say, pruned.
So, okay. So microglia, we've come to appreciate in just, the past one generation of scientific research that they're doing a lot more than we used to think they're doing. And at least some of that has to do with being involved in the synaptic pruning that's happening at a massive level. That's very important, especially at certain stages of brain development.
Yeah. So, I mean, I think a lot of this emergence is still there's still some good scientific debate as to the active versus passiveness of this process. And
Hmm.
so when we say pruning, that is an active process. The microglia are physically removing or clearing those connections, whereas phagocytosis, although it's requires energy, they have to eat things.
Yeah.
It's still passive in the sense that they're not physically killing the neuron themselves. They're
Right.
taking that up. So there are still areas that, such as that are controversial and I think really important, areas of scientific debate. But I think it's very clear that there is this importance for the ability to clear at least those dead cells. And I'll just add one other interesting area of scientific debate, I think, is um, redundancy. And we talked
Yep.
about this,
Yep.
biological redundancy, where there's a series of emerging studies in mice that suggest that if you get rid of microglia genetically or pharmacologically, that you can still generally develop those animals okay. And that's because perhaps astrocytes or other cells in the brain can assume the responsibility of that clearance. And I always like to point out that, like, I think both sides are probably correct.
Yeah.
And
Yeah.
really what it is just that whenever you completely eliminate one cell, you have redundancies in place.
Right. And I mean, that's been observed in many, many areas of biology, right? Like you delete a gene and nothing happens. And some people will mistakenly conclude, oh, well, that gene wasn't doing anything important. It's like, well, actually, it was doing something so important that there's a backup.
Yeah.
There's a backup system.
Yep. Whereas if you were to just manipulate or modify, like we do, where you still have the cell in place and you just sort of modify its normal function in a way that is, measurable, then you see those phenotypes emerging.
Okay. So let's now step back and talk about sort of fructose stuff at a higher level with respect to human beings before diving into the paper that we'll dive into. Fructose availability and consumption over time. Just give us a lay of the land here. I don't know, for the last 50 or 100
Mhm.
years, what is fructose consumption for the average, let's just say, American or person in the Western world looked like? Has it gone up? If so, by how much?
Yeah.
Fructose exposure, what is that looking like in people?
For sort of the last several generations. I mean, it's particularly with the addition of what is a cost-effective additive from the high fructose corn syrup is just skyrocketed astronomically, right? I mean, you have huge increases. I mean, I could point you to any one number of industrial studies. You could even go look at, there's a pretty famous study looking at World War II, pre, during, post World War II in the UK, where they had sugar rationing and then the use of fructose after the fact. I mean, it's astronomical the amount that we've used. And I always like to point out that, I don't want to ascribe motive to why people did what, but there was a general benefit to malnutrition, the ability to have a cost-effective additive in this case, right? It was a cost-effective additive. However, unintended consequences, this is like classic human nature, right? Is a solution to something led to sort of catastrophic consequences, and many of those are related to obesity and diet related, and a lot of that, I think more and more too, it's not just fats or, I think it's, we're hearing a lot about ultra processed foods, but I think a big component of it is going to be this massive increase in fructose consumption because of what we talked about.
Mm-hmm.
Fructose, unlike glucose, there's not a lot you can do with the fructose other than making lipid or converting it to glucose. So you have
Yeah.
double
Yeah.
fructose, right?
So most of our cells can't utilize fructose for energy like they can with glucose, but fructose, particularly in the liver, can be basically used to make more fat. And so that you start thinking about fatty liver disease and stuff like
Absolutely.
that. But another thing that connects into this, I think, is that so not only is it economical, so it's cheap. That's one of the reasons why it became so widely used. It also, when you play with the glucose fructose ratio, and usually when you bias yourself a little bit towards fructose, this changes the perceived sweetness and palatability of food. So your motivation to continue eating something because it tastes yummy is also fructose very much is important for that palatability tuning.
I 100% agree. I think the taste is key. I mean, often, in a former life, I worked with rats, training them to be alcohol preferencing rats, and one of the things we would add is fructose, right? You want to make it taste good. You want to make them come back for more, and it's not just the alcohol that is absolutely addictive. It's also that fructose additive that you add.
Mm-hmm. And so what do we know about high fructose consumption? And I'll let you define what that means. Higher than average, how high is high? We'll get into that stuff. What does high fructose consumption correlate with strongly that we think makes it worth studying
Hmm.
under the hood? Like, is high fructose consumption actually correlated with bad stuff clearly?
Yeah. So, I mean, I can talk a bit about the areas that I'm not an expert in, but I think are generally well consensed, such as you mentioned it, fatty liver disease and consequences of these sorts of inflammatory and chronic liver diseases, as well as, obesity, et cetera. Um, you had a wonderful guest on that talked about its contribution to cancer development. And in fact, colleagues here at Memorial Sloan Kettering, where I'm at, have also seen similar things where, fructose can contribute to leukemia development. So leukemic cells, unlike normal stem and progenitor cells in your bone marrow, those leukemia cells can actually use that fructose. So that's a rare adaptation that they're very good at doing. And that's because fructose can be high in your serum, in your blood. Um, and then, where I come into this particular discussion is again, as a trained psychologist, I became really interested in the epidemiological, I'll call it a correlation between, this even pre-COVID, but especially post-COVID in not just the United States, but in the much of the Western world, huge increases in developmental, what we call neurodevelopmental diseases. So
Mm-hmm.
diseases, mood and anxiety disorder development, autism, spectrum disorder development, even above the diagnostic increase, right? Like even though we're better at diagnosing things, there's still increases across the board in a lot of these. And a lot of really striking and strong correlations with this increase in fructose consumption. And in fact, fascinating studies showing that even and we'll get into this, but even a single, you give a pregnant woman, a glass of juice, you
Mm-hmm.
can actually measure the fructose in the breast milk after a single glass of juice. You
Hmm.
can measure that spike in fructose in the
Hmm.
breast milk. So this sort of correlative and suggestive data beyond the thing we know about fatty liver disease really got us interested in you know what could this be doing to neurodevelopment.
Hmm. So does high fructose consumption in either pregnant women or young people, children, does it correlate with neurodevelopmental issues?
Yeah, and it quite strongly actually, and I think there's a growing consensus on um, I say correlation and they do causal modeling, but really there's never been, you can't do that, right? Like that
Right,
would
right.
not be
They like,
particularly ethical
yeah.
to do.
Yeah.
And of course, I want to just make clear too that though in psychology it's called a two-hit model, that
Mhm.
is you have a combination of susceptibility and a triggering event or triggering events, right? So classic example would be, for schizophrenia is you have overbearing or prodromal symptoms or an overbearing mother combined with, let's say, a traumatic event or something happening that might trigger, that disease. And so for us, it was a lot of the evidence suggested that it's increasing the susceptibility
Mhm.
to these diseases, and it's both. So it's both in pregnant women, the offspring of pregnant women, as well as adolescents too, who
Yeah.
consume
Yeah.
fructose.
And just to give people some sense here. So I wrote an article about fructose some time ago. I did a back of the envelope calculation just using numbers that are published out there. We know, I mean, because we know how much sugar sweetened beverages out there. Like you can get a pretty good calculation of how much fructose, say, the average child is consuming today. And then, like, it turns out wildlife ecologists know how much fructose chimpanzees eat. And normally when we think of a chimpanzee, we think, okay, it's an ape living out in the world in the jungle and they eat bananas and fruit all the time. And bananas and fruit are some of their diet. They eat a fair amount, but within a certain margin, children today are probably drinking, a similar mass of fructose every day that a wild chimpanzee is
Mm.
eating. And, they're not climbing trees to get it from whole fruit. They're drinking it from sodas and juice boxes. So
And
the sugar load that people and young people are exposed to today is very high.
And just to add to that too, there's some really beautiful work from a group at Princeton, Josh Rabinowitz and their group, that has also shown really nicely there's actually a distinction between liquid and solid
Hmm.
food consumption. So you just actually highlighted,
Yeah.
you know, fruits, berries, et cetera. That's a solid food consumption
Right.
versus drinking a juice box or a soda,
Right.
et
Right.
cetera, or breast milk consuming
Right.
liquid.
Right.
And so what that group has showed through a series of really nice studies that the way we metabolize, transport and consume, it matters, right? So if you're eating solid fruits, vegetables, et cetera, you're getting it through solid food. The impact on your liver and then
Yeah.
presumably on your brain
Yeah.
is much less than if you're doing it by fluid,
Right,
by
right.
liquid.
Because, all of the dietary fiber and solid stuff is limiting the rate of absorption in your body. And so effectively a liquid fructose concoction, whatever it may be, it's effectively going to be a higher dose or potency than something eaten like a solid fruit, because you're just going to sort of, you're going to be hit with more fructose quicker with the liquid concoction.
And Josh's group suggests they use the shield analogy, right? That the combination of your small intestine and your liver, but especially your small intestine, is a shield to high fructose. And if you consume it too much in this sort of liquid form, it overwhelms the shield, right? It breaks through that shield.
Mm-hmm, mm-hmm. And then just to sort of repeat one more time something you said before, because I think it anticipates some of where we'll go. Even before weaning, when a little baby is living off its mother's breast milk, if the woman is drinking whatever, juice, soda, the fructose, you can detect the fructose in the breast milk. And so the effects of high fructose consumption by the mother will actually pass to the child through the breast milk. Is that basically what we know?
Yeah, and just take a minute to think about how profound this is because, if you talk to now obviously there's medical guidance and then there's, sort of standards and whatnot with obstetricians. But, we do or they do screening for gestational diabetes as late as the second trimester, right? So essentially, if you have you know, they're basically concerned with those first two trimesters of gestational diabetes, and then at that point, they stop screening it. But what a lot of this suggests is this breast milk is, first of all, you have third trimester of development, and then you have just post pregnancy, right, where you're still exposing. And so if you're suddenly consuming fructose, high fructose, drinks, et cetera, during that period, you're still affecting their development, even though you've made it through those two early trimesters that are absolutely critical. But you've basically, I'm safe. I didn't, I never got gestational diabetes. I can have
Right.
these sodas,
Right.
et cetera. Yeah.
Interesting. Okay. So we've set up a lot of the pieces here. Walk us through this new paper. What was the setup here in terms of why you did this and how you designed some of these experiments? So we're going to be talking about a paper called "Early Life High Fructose Impairs Microglial Phagocytosis and Neurodevelopment." So we've sort of alluded to where we're going with this, but what's the setup here for these particular experiments?
Yeah, so I'm happy to also give sort of the background of also why we were interested in this for sort of the evolutionary context
Hmm.
too. But
Yeah.
it's basically I read a paper on naked mole rat. Naked mole rat are a species, rodent species that violate a law of nature, which is they live very long, 20, 25 years, and they do so by using fructose instead of glucose for their metabolism. They do it in low oxygen. They can live in like no oxygen environments or very low oxygen environments, and they do it by using fructose through this transporter SLC2A5 glu5. And we've already alluded to that the microglia are the only cell that have this. And so, and based off of all of this evidence of the high fructose diet in pregnant women and in adolescents, So we basically designed, first and foremost, asked the simple question, does high fructose diet, and we can talk about why ours is a physiologically relevant high fructose diet, but does a high fructose diet affect those microglial phagocytosis, in basically in the neonates, right? So if you give that fructose diet to the pregnant dam, what we call pregnant mice, does it transmit and affect the microglia in the
Yeah.
neonate?
Uh, Maybe I just missed something. So naked mole rats, they live a long time. I already knew that. People have different ideas for why. What's the connection between that creature and fructose? How do they eat fructose?
It's extremely fascinating. So every one of their cells, pretty much, or all of those tissues, have this transporter, and they can use fructose. There's,
Where do
we're
they
still,
get fructose from?
yeah, that's a great question. So they do insects, actually. So
Hmm.
they get it from the insects they consume underground because they are underground dwellers, and so that's a major source of their fructose. And they are, there's some interesting biochemistry there. Basically, the rate of metabolizing fructose is a little slower, and they have slower metabolism. But
Hmm.
all of this was just basically, I was just really curious, and so we designed those experiments to try and test this really striking observation that unlike them, mice and humans only have this transporter in certain places.
Right, right. And the place that we have it, mice and most other creatures probably, is in these microglia.
Is in these, yeah, in this microglia. It's
Yeah.
extremely striking.
Yeah.
Yeah.
So we've said microglia, these are the resident macrophages of the brain. They are involved in cleaning up trash in the brain. That's very important from a developmental standpoint because so much pruning and clearing needs to happen for proper brain development. They, unlike other cells, have this fructose transporter. We know from the epidemiology there seems to be a relationship between high fructose intake and problems that people have. And so some of the pieces are in place for you to say, okay, well, maybe high fructose exposure early in life is doing something to these phagocytes, and that's impacting brain development and subsequent behavior.
It perfectly set up. That's exactly right.
So walk us through the initial experiments here. What did you actually do?
Yeah. So one of the things that we found is that a lot of groups who do high fructose studies use a super physiological amount of fructose.
Yeah. How much is that?
It's like 60% by weight, essentially, of the chow or volume.
Oh, so of all of the food. Yeah.
Yeah. So it's quite so. Whereas ours was about 10%. So
Mhm.
ours is literally the equivalent of in humans would be about a can of soda a day. So an 8 ounce can
Okay.
of soda a
So
day is
yeah,
what
so not even someone who's just like binging soda all day every day, just one can of soda per day, which is going to be, I mean, what are we talking about here in terms of high fructose corn syrup? Is that about a 55-45 mix of fructose to
exactly right.
glucose?
Yeah, exactly right. But of course that's not what all of the soda is made up of, right? So even though
Right,
it's
right,
55-45 fructose, there's a lot of other
right,
stuff, good or bad,
right,
in your
right,
can of
right.
soda.
So it's equivalent. So the dose of fructose you were using for these experiments is equivalent to about a can of soda for a human per day. So this is not an insane amount.
No, no. And in fact, I
But
figured if we see any phenotype in that, like that's going to be even more meaningful, I
right,
think, the
right.
study. Yeah.
Also,
And
another way of saying all that is a lot of people are consuming way more than this per day.
way more than this, way more
Yeah.
than this. And also the other reasoning was this was a volume contribution during pregnancy, but I'm not even taking into consideration that the solid food, if you eat too much solid fructose, you can still have this effect, even if it's
Right.
not as efficient as causing issues as the liquid forms are.
Okay. So walk us through some of the first results here
Yeah.
that you got to.
I mean, and so the way we read this out is looking for uncleared dead cells and or accumulation of neuronal material in microglia. And it was quite profound, actually. So when we looked at mice at 7 days after birth, so what we call perinatal day 7, if you look at the prefrontal cortex, so the in the developing mouse, about estimated between 70 and 90% of the brain during this early period is the cortex, is that frontal cortex. If you look specifically at that region, we saw huge increases in uncleared dead cells, meaning that they weren't doing their jobs, and also they were accumulating less material inside of them. So they were
Hmm.
doing less
Hmm.
phagocytosis.
So less is being cleared out, and it really looks like the phagocytes are just not cleaning up as much as they should be.
Exactly. And yeah, I mean, that's exactly right.
Mhm. And so remind me, so for the entire pregnancy, the females are getting this high fructose diet, which is not an insanely high fructose diet.
And bear in mind too that we went through painstaking effort to try not to keep mice who pregnant mice um, too long on these diets. So this diet was they were only on this diet for 3 weeks or so during the
Oh, okay.
pregnancy, right?
Okay.
So because you don't want artificial effects of obesity, et cetera,
I
right?
see. I
And
see.
so we
So
controlled for yeah, yeah.
I see.
Good.
So you're boosting their fructose intake, but not enough to give them obesity, which could cause a host of other things to change, which are effectively
Yes.
confounders for you. And again, it's not an insanely high dose of fructose
Correct.
here. This is a
Yep.
very reasonable amount.
Yep. And they're not gaining more weight than their normal diet.
Hmm.
And so that was one aspect. And then the other way we really tested this, two other ways I think are really worth mentioning. The other one is we actually then were curious if the effect was directly on the neonate or something occurring during the pregnancy.
Hmm.
And so we did this experiment called a cross-foster experiment. So basically, the simplest way to explain this is where if you have, high fructose pregnant mouse gives birth to a litter, you take that litter and put it with recently pregnant normal diet mouse so they can breastfeed and wean with a normal diet. And then
Mm.
vice versa. You take mice born to a normal diet and you put it with the high
Yep.
fructose diet. And so I thought one or the other would give us a significant difference either way. And it turned out both of them still cause a phenotype. So the
Huh.
defect is occurring both during the embryogenesis, so in the womb, and also after the fact. And so, yeah, and then I'll just say the secondary way we confirm this is we also gave a tenth of the amount of fructose, so even less amount, directly to those neonates. So during an early day 2 to day 4 post birth,
Yeah.
if you just give them 1/10 of the amount of fructose into the stomach, you still see the same exact phenotype giving it to the neonates. So even less fructose, if your young developing
Wow.
children are doing this, there's still that effect. So those are the sort of experiments, big ones.
Wow. So fructose exposure during pregnancy, in the womb, or afterwards, even if it's quite a small amount, is causing this failure of phagocytes to clear out stuff in the developing brain. So then there's too much stuff in the developing brain.
Absolutely. Yeah.
Wow. What comes next in this story?
Yeah. So I mean, this is where we were curious if this was the classic. Is it cell intrinsic to the microglia, or is it something happening on the outside? And so through a series of experiments, we basically made new mice, a whole new mouse strain where we could specifically delete the transporter that I mentioned, GLUT5,
Yep.
from microglia. Actually, it deletes it from all macrophages, and only in the neonates, so in the
Mm-hmm.
offspring,
Mm-hmm.
not in the
Mm-hmm.
mother,
Yep.
right? Because if you're being honest, it could have easily been a phenotype that's still occurring in you know not in those microglia. It could have been a
Right.
small
Right.
intestine issue, it could have been a liver issue in the periphery, et cetera.
Mm-hmm.
And it turned out that those experiments where it's only deleted from the microglia completely reverse that eating
Mm-hmm.
phenotype.
Mm-hmm.
So those cells were completely able to eat.
I see. So all of the fructose induced problems that you saw in the developing brain, the failure to clear out all the gunk, required intact fructose transporters in the microglia of the little baby mice.
Yep, that's correct. I want to be clear that, there probably were also peripheral effects still
Yeah.
happening on the neonates, on those small mice. Um, we just didn't look at those, but at least when you only hone in on the microglia phagocytosis defects, those were specific to the fructose. The effect of fructose was specific to them.
Mm-hmm, mm-hmm. And what's sort of like implied in a way by some of these experiments is that, you said you could get this phenotype even if there was no fructose exposure in the womb, but you just gave them 1/10 the dose shortly after birth, that starts to imply things like, oh, a small amount of fructose in, say, the breast milk of a human could potentially go in this direction.
Exactly. It implies that, I mean, if you don't want to like hope for a result, but I was kind of hoping that it was reversible because that would have
Right,
important public
right,
health implications. And it was not reversible. And so that was just pretty striking to me was, you know, that those hits, those early hits make you susceptible. And we'll talk about what the ultimate phenotype, behavioral phenotype was. But, those early hits, they have their damage and it doesn't seem to be reversible just by stopping.
right, right, right. And so, we see what happened at the cellular level? There's a phagocytosis deficit. Stuff is not being cleared out in the developing brain, in the frontal cortex in particular. This comes with just a fairly modest amount of fructose, either in the womb or even just after birth. What happens to these mice when they grow up? How do they look or behave differently than normal mice?
Yeah, so I think that's a really important question because, just because you have this cellular and physiological phenotype, you want to know if it's affecting behavior, cognition, etc. And it's hard to do in mice. However, there is a well documented, well understood neural circuit that underlies fear conditioning. So fear is highly evolutionarily conserved, and it's pretty much same regions of the brain in multiple organisms, and for us and for mice, it's basically links. We talked about the prefrontal cortex linking to the amygdala. So this is a classic fear circuit. And so just to explain what this experiment is, it's very classic. So, like as in like, classic B.F. Skinner and classic behaviorism. So you put a mouse in a chamber, and that is an electrified floor. So I don't want to imply like we're not doing tons of damage to them, but it is enough of a stimulation that it invokes a fear response. And so when you pair that shock with either a light or a sound,
Mhm.
they associate the oncoming of that shock,
Yep.
right? The stimulus to the light or the sound.
Yeah, it's like Pavlov's dog. So the next time they
Exactly.
hear the bell ring, they freeze because they think something bad is about to happen.
They think something's going to happen. Yeah. And we know that that particular circuit isn't a memory thing, right? Because of just decades of research on this circuit. And so it turns out, though, that if you then come back a couple days later, you put those same mice that have been conditioned to fear that in that chamber, and you start doing the sound or the light, but you don't shock them. So their initial response is to freeze.
Yes.
Mouse's fear response is freezing. And over time, it's called extinguishing. You extinguish that freezing behavior. They'll no longer
Yeah.
respond
Yeah.
to the stimulus with that fear response.
Yeah.
So
So
it's classic.
initially, they very quickly learn, oh God, that sound means I'm going to get shocked. They freeze. They learn that very quickly. But then if you expose them to that sound, but no longer it comes with the shock, they eventually thought, okay, it's okay.
Yes.
I don't need to worry about this anymore.
Yeah, and over a series of trials, within a day, they'll basically return to where they're just moving around normally, like nothing happened.
Mm-hmm.
And so that's what we call fear extinction. Yeah.
So mice learn this, they unlearn it. There's sort of a known rate that this happens at. The circuit is very well characterized. This is just a classic way we can measure things like cognition, learning, memory, and behavior in an animal like this.
And I'll make this more relevant to humans. The reason why this circuit is so important for things ranging from PTSD, which is obvious, right? You're pairing, like you're a soldier at war and you have, bomb noises paired with violence, and then you go home and you have a firecracker or a firework, right? That's a classic example of that. But it's also observed in patients who suffer from mood or anxiety disorder. It's not a violent pairing necessarily, but perhaps someone pairs a robbery or something happening, some negative evoked response. They pair it with a place, time, objects, people, etc. And those pairings, even though nothing is happening to them, that can lead to these what we classify as mood or anxiety disorders.
So learning and unlearning are very critical for people and animals. And many of the things that we consider to be disorders or to be problematic, whether it's PTSD, whether it's anxiety, whether it's very severe or more mild, many of them have features that we recognize as symptoms that look a lot like the failure of the brain to unlearn or extinguish something that is no longer relevant.
Absolutely. And I'm obviously of a specific school of thought on behaviorism and cognition and learning, but at least that's the principle of a lot of therapy too, for what it's worth, is CBT, cognitive behavioral therapy, is really the process of unlearning learned behavior.
Right. Yeah, the therapist is helping you extinguish things that don't need to be paired together anymore.
Yeah. So this is an important thing, and I can walk you through the result, what we saw. Yeah.
Yeah. So the fructose exposed mice, what do they look like?
Yeah. So if mice born to dams on normal chow or normal diet, they are able to extinguish this. We observed two things. First of all, mice born to dams on high fructose have even higher freezing response just from the get-go. And they were almost completely unable to extinguish that fear response over those trials.
So they're more afraid, and then they stay afraid.
Yes, yeah, exactly.
And what did you do? Any other types of tests, or any way that they see any other ways they look different?
Yeah, so I mean, we, as is typically the case for a lot of these, you do other behavioral and memory and other batteries, and so we actually looked at things like spatial memory, working memory, a lot of the classic tests, one's called the Barnes maze, these, ability to remember where a hole is and escape. And we didn't really see any memory defects. But one other interesting thing we did observe was a defect in what we call the novel object recognition test. And so just to simply explain this one, because it's kind of an interesting one, is you put a mouse into a chamber with two objects in there. So they're the same object, right? So it's a blue square or something, and let them explore and roam around. Then you take one of those objects away and replace it with a red ball. The mouse, because they like novel objects, will go explore the red object. And so the issue with this test is that it tests two things. One of them is cognition, the other one is memory. And so do they remember the thing, and do they cognitively recognize that those things are different, right? And so what mice on high fructose, I just told you that they didn't have any other defects in any other memory battery, but what they do is they actually fail to spend time with the novel object when we put that in there. And so what this suggests is that it's more support for our interpretation of there's fear behaviors, right? So they don't want to observe the new.
Well, I think, I think most people or most people in this realm who study animal behavior like this. So prey animals like mice, right? The term here is neophobia. They're usually a little bit skeptical of new things until they get used to it. And so that's one way to interpret this, right? Is that this is sort of a high anxiety, high fear phenotype. And in addition to the fear conditioning result you just told us about, there's also this way of interpreting the novel object recognition. Like they're not going to the new one and checking it out as much as a normal mouse.
Absolutely. And as long as, and like I said, paired with the other batteries that are actually assessing only spatial memory, working memory, etc. And none of those had those defects. Right.
So they can still figure stuff out, but they're basically just more skittish all around.
Correct. Yep.
Yeah. So how do you interpret first of all, are there any other major experiments to go through here?
Yeah. And so going back to the mice we made that specifically deleted that transporter from microglia, it turned out that similar to our cellular phenotype, these mice also had complete reversal of the behavioral phenotypes when we got rid of the transporter from all macrophages, but particularly microglia.
So just to make sure I have everything here. So when you get rid of the fructose transporter, the microglia can't uptake fructose, and these mice perform like normal mice on these tasks. In the case where you've deleted that transporter from the microglia, and they perform normal on these behavior and cognition tasks, Is there still a clearance deficit at the cellular level? Is there still stuff not getting cleared out?
So in the where we've removed the transporter,
Yeah.
it completely recovers their capacity to perform this clearance
Okay. Okay.
process.
Everything goes back to normal, basically.
Everything, well, I guess that's ultimately the question, right? Is is
Well,
is
the
there
stuff.
an issue with deleting the transporter long
Right.
term? And
Right.
that's, I
Right.
think, the
Right.
next stages of the investigation is, okay, this is fine for this, sort of, because remember, high fructose is not supposed to be a normal situation.
Yeah.
We're
Yeah.
reversing a non-normal situation, but are we really making it normal, or are we just reversing a bad situation? I can't answer that. That's the
Hmm.
next thing, is to try and figure out is, okay, now these mice are born without the transporter. What happens to them either during later stages of development or
Yeah.
during healthy aging, et cetera? And
Yeah.
I think that's a really interesting question we're trying to get at.
Interesting. So sort of overall, how do you interpret these results in light of the stuff that people care about, the stuff that we talked about before about, fructose exposure going up and it being correlated with all these negative neurodevelopmental outcomes in people? How are you interpreting your results here? And what do you think this implies for what you look at next?
Yeah. So I mentioned that I think the safe way to look at this, I mean, it's animal studies, right? We always have to add that caveat, but I do think that this is a strong indicator that our diet, particularly high fructose consumption, um, is a really important contributor to susceptibility to developing these sorts of diseases, right? So it's like fructose alone, I don't think will cause, you know, any of these diseases. But you're definitely playing with fire as far as it goes, right? Like, I
Mm-hmm.
think it's a major contributor. And, I think probably everyone who's listening to this is familiar with the work on, smartphone use and a lot of these other things that are also thought to be associated with the rise in psychological disorder development. So you can imagine you're already taking generations of kids and making them more susceptible,
Right.
and then pairing it with the trauma of lockdowns or the trauma of, you know, TikTok or whatever, choose your favorite, choose your own adventure in that
Right.
case.
Right.
We're basically making the case that this is, lowering that threshold for the development.
Yeah.
Not everyone will.
Right.
And there are other
Right.
reasons, there are resilient reasons why kids won't develop these things. But it is, we think, is a contributing, the
Yeah.
susceptibility.
But through developmental mechanisms like this that lower, say, the threshold for susceptibility to various, what we'll just call environmental insults or aspects of modernity that can have negative effects. There's 1,000 things out there that could have those negative effects, such that if you lower this threshold, any one of them or a combination of a small number of them can now, let's just say, push you over the edge or push you in a direction where you're not going to be as it's going to have behavioral effects and cognitive effects ultimately.
Yeah. Yeah, and not everything has to be pathological,
Right.
right? So
Right.
a disorder is something that has a pervasive detriment. That's the DSM
Yeah.
definition of a disorder,
Yeah.
is something with a pervasive deficit on your life, day-to-day life. But that doesn't mean that the many of us can't
Right.
have something in between,
Right. Yeah.
health and disorder, right? There's a
Yeah.
lot
How do we
big.
know that we're 10% more anxious than we would have been?
Yeah, I mean, and you just the ramifications are, dating and mating and
Right. Right.
relationships, and just you can just think of the areas where this could be and I just want to add one other thing that we think about a lot is, so in mice and rats, that developmental window, that period, what we call a critical window where this can have an effect, is short. It's only a week, 2 weeks, maybe three. In humans, it's over 20 years. Some estimate up to 25 years. So, but at least the first 18 to 21 years of life, you are susceptible to these sorts of
Right.
modifications in microglia function and neural pruning and neural turnover. So our window is much, much bigger than
Yeah.
rodents.
And so just to reiterate some of the things you told us before, right? You saw these effects from fructose exposure with relatively low doses of fructose that are some comparable to the amount that the mice got is comparable to what a human might be exposed to by eating just or drinking one soda per day. So not an extreme amount. You used even less than that when you exposed the little pups after they were born instead of during pregnancy. And we know that high fructose intake by a mother can be passed on via the breast milk. So when we think about things like dose here, sometimes, people get into debates, obviously, about high fructose corn syrup, and they say, well, it's got more fructose. And then people say, well, but it's only 55-45 and table sugar is 50-50. That's only a 5% difference. When you think about all of those things, like how high is too high? Like is a 5% difference potentially consequential here based on what you've seen in your experiments?
I'm going to be honest with you. I probably wouldn't even use regular sugar drink if I were pregnant, right? I mean, because I understand the argument that a 55-45 versus 50-50, but like any of that fructose during these, it's probably not great, right? And so, I think you made the point of sweeter, more consumption, more addictive. All
Right.
of those things are
Right.
extremely relevant in this
Yeah.
conversation. But I think
And
just generally a sugar versus non-sugar drink is itself already,
yeah. And this is where, in my experience, as a person who kind of does a lot of this and interacting with people who care about the stuff, a lot of people get tripped up when we talk about things like this that seem subtle, but they're not actually considering all the synergies and second order effects that can come from this. So what you just implied, I think, is if you have high fructose corn syrup, let's say, compared to table sugar, you're talking about only a 55-45 versus a 50-50 mix. But the enhanced palatability of that 55-45 will probably also induce those individuals consuming that instead of the regular sugar to consume more overall. So there's a lot of these sort of interactive and second order effects. And especially when you think about developmental sensitivity and you think about the fact that we're not talking about one meal here, we're talking about like daily consumption habits. These things stack up and probably do move you in one direction compared to another.
I mean, it's let's put it in real terms, right? We're not talking about one can of 50-50 versus 55-45.
Right,
We're
right.
probably talking about two cans of 55-45 versus one can, of 50-50
Yeah.
or whatever that
Every day
exactly what you
for
said, right?
weeks or months or years or whatever. Yeah.
Yeah. Yeah. Or even, I get asked this question. You never want to give medical advice, right? But I'm happy to basically say, like, if you're at all questioning whether or not I should even have that one bottle, 20-ounce bottle of Code Red Mountain Dew. I say this because my wife had kids and, I've been through this. I probably wouldn't do that. I'd probably recommend don't even take that risk, right? Because we just don't know, you know, we just don't know enough about like what even transient exposures, right? Like we haven't even in mice,
Right.
we haven't done that experiment where it's give it, you know, once a week instead of constant chow, right? Or give it a
Yeah.
couple of doses during a
Yeah.
certain period.
But you said also you didn't give it the whole pregnancy, right? You still did give a fairly limited amount.
Yeah, and that was, in my mind, really important to say. But sometimes you'll hear the advice where like, oh, my doctor told me to only do, like I can
Right.
have this
Right.
occasional. I
Yeah.
don't even know if I would take that chance,
Right.
right? This
I mean,
doesn't
going
seem.
back to your own experimental setup, that I think you said this too. Like you gave only a modest dose of high fructose. You gave it for a limited window in the pregnancy, and you gave an amount such that the females were not gaining extra weight or showing signs of metabolic macroscopic
Yep.
obvious signs of metabolic
Yep.
disruption. So they looked fine.
They looked
They
fine.
seemed
Yeah.
healthy.
And that was an important, I mean, the scientific process, you have reviewers criticizing it. And that was an important criticism was just show us what the weights are. And I think it was an important piece of data kind of buried in the supplement somewhere. But if you see, our mice were not gaining any more or less weight and they didn't have different metabolic metrics. So yeah, that's a great point.
Yeah, yeah. Interesting. Um, okay, so where do you guys see this going next in terms of your own work?
Yeah, there's two angles. Well, three really, but one of them is I mentioned you know, we our microglia have this transporter. Uh, you know, evolution doesn't uh, do things for a reason, right? There's no intention behind evolution. So it's entirely possible that this is just something that's remained behind and doesn't have a purpose. But we hypothesize that it does. And so why is this transporter expressed in microglia at such high levels, particularly in it? So it turns out the expression of this transporter in humans and in mice goes up with aging.
Yeah, yeah.
And
Interesting.
so the question is, does it serve a role? Does it actually do anything? I told
Yeah.
you we've now deleted it. What happens to
Right.
that
Why
organism,
is
right?
it there in the first place? What's it
Why is it
doing?
there in the first place? And I get asked this question a lot, and I think it's a really important question. I have weird speculations about hibernation and maybe it's keeping. So, right. So we talked about how microglia they do this phagocytosis early in life,
Yeah.
but then after these development windows, they sort of go quiet. I don't want to say they're like quiescent or completely quiet, but
Yeah.
they do much less
Right.
phagocytosis.
There's less for them to do,
Less,
ostensibly.
and in fact, it's sort of a Goldilocks model, right? Like you need them to do enough, but you don't want them to do too much too.
Right. 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 Of course. Yeah, that
And
makes sense.
so the hypothesis is that maybe this is sort of how Goldilocks ensures the appropriate temperature, right?
Right.
So that not too hot, not too cold is through the limited use of fructose through this transporter in
Yeah.
adulthood and aging.
Yeah. And where my mind goes is if we think in terms of, seasonality and animals like rodents that often go into things like torpor for part of the year, I wonder if there's something to, their metabolism has to change when they go into those states. And I wonder if somehow this has to do with that.
100% agree. And this is where we go back to our friend, the naked mole rat too,
Right.
right?
Right.
Is their capacity to live for 20 to 30 years must have to do with their ability to use fructose in a unique way. So it's not the brightest idea, right? It actually kind of just makes sense that maybe that's why your microglia, which you and I probably still have 95+% of the microglia we were born with, maybe it makes sense that they are using this fructose in limited amounts for a purpose now.
Yeah. Yeah. And, I don't want to dwell on this. We could probably have a whole podcast on this, but something that I've started to notice as I've scanned, part of what I do is, I'm not in the lab like you are, but it's almost like I can see the forest instead of the trees compared to
Mhm.
the working scientist, because I sort of learn a little bit about all of these different subjects rather than go as deep on one of them as you guys do. I'm noticing a lot that there's a lot of disease states and disorders which seem to do with phagocytes getting sick, whether it's macrophages getting poisoned by oxidized LDL when we think about atherosclerosis, whether it's a whole host of brain stuff that seems to keep coming
Yeah.
back in different cases to, I'll just call sick microglia. These things not only cause problems, but they can actually travel to other tissues and move the problem around. Even in things like obesity, like there's a lot of macrophages that seem to just get wonky and messed up and cause inflammation and stuff in the adipose tissue. Do you think that phagocytes are a sort of, of outsized importance in terms of, when they start to get sick, the body starts to get sick?
I mean, this won't come to a surprise to you, is that the macrophage phagocyte biologist says that there's an outsized importance for phagocytes in these disease processes. But I think you're not wrong. And in fact, my lab doesn't just do microglial development. We agree with your worldview, where we basically want to know what all phagocytes do in all tissues,
Mm-hmm.
because, as an example, lysosomal storage disease proteins, the vast majority of them are actually restricted to macrophages, to phagocytes.
Mm-hmm.
Some are in neurons. Some are important for neurons and autophagy, the self-eating process. But the vast majority of them are in phagocytes, and their lysosome storage disease means like lysosomal processes. So intuitively, it makes sense that a lot of these, what we call inborn errors of development and inborn errors of neurodevelopment are probably, we think, and it's a major area for us, is inborn errors of phagocytosis. The other place you mentioned athero, I love this one because one of my colleagues is a world expert in athero on the phagocytosis in athero. And even he's come around to this idea that early on during atherosclerosis development, it's not macrophages not clearing stuff. It's that they're clearing stuff and they can't digest the lipids. They can't digest
Yeah.
the and then they begin forming those plaques, and then
Yeah.
eventually you get the necrotic core, and eventually you get the damage issue. But
Yeah.
everything early on is all coming back to this digestion issue,
Right,
right?
right. And just if things become too poisonous, basically, like these cells are designed to handle nasty stuff, but everything has a limit. And if things are just too nasty, too much, the fact that they're handling it means they're going to be exposed to it even more than other cell types, probably. And so it seems very natural to me that these cells could be critical both in terms of their sort of susceptibility or the probability that something goes wrong just because they handle toxic stuffs, by their nature. And also they move around. So they can then, if it moves from one place to another and then that leaks out, like there's just lots of points of problem that can arise here.
And you just alluded to what one of the most important aspects of this in my mind is, we can also take this in the context of cancer, because we obviously also do cancer work. Macrophages, phagocytes, but macrophages specifically, unlike a lot of the other immune cells, are the cells that are always there, and they can get into tissues and they can go the places you need them to. So that's both a problem, as you note rightfully, but it's also the solution in my mind, right? And so this is an area of our work and my colleague and I's work is engineering. Can we engineer these cells to either reverse the bad thing they're doing, or can we give them the capacity to do the thing we want them to do? And this is a very exciting area of research, is because these cells can get to the places, you imagine a tumor, you have all of this collagen and these like tight tumors. T cells can't get in, immune cells can't get in, macrophages can. And so if you can engineer your macrophages in a way that suits anti-tumor effect, or can clear lipids better in the plaque, or you name it, go to the brain and help clear. I mean, I'm never going to argue that we should do transplants to children who are on high fructose. That's maybe not a good way to do it. But if you can understand how they're supposed to be doing this digestion process, can you maybe counteract the negative effects of the fructose by enhancing the appropriate activity in a safe and healthy way, right? So instead of trying to get kids to not consume fructose, which is very hard, can you maybe add another thing, a nutrient or something else that can counteract the negative effects of those fructose? I don't know if that's possible, but that's certainly how we're thinking.
Any final thoughts you want to leave people with, or anything you want to reiterate from our discussion, especially maybe for those who have or will soon have small children to think about?
Well, I when I was in psychology, I remember my kids were very, very young. I think they were 4 or 5 when I started. And I remember asking my professor, the CBT, pediatric expert, what should I do as a parent? And he said, I don't know. I can tell you all the things you shouldn't do, but I can't really tell you what to do. So, there's a lot of information out there. Ultimately, all you can do is do the best you can. And so, I think limiting fructose is not an unreasonable thing, but you're never going to keep a kid from not consuming fructose. And so there's just a lot of other things that you can do to enrich kids' lives and to do these things. But I think, just do the best you can ultimately, because I know I wasn't perfect at raising my kids with not consuming fructose for sure. And I definitely consume fructose myself. So that's all I would say.
All right. Well, Dr. Justin Perry, thank you very much. This was fascinating.
Thank you so much.
Visit mindandmatter.substack.com to find all of my content. Hey everyone, I want to take just a minute to tell you about one of my cool new affiliate partners called Seed Oil Scout. Seed Oil Scout has a really cool map feature that helps you find restaurants in your area where you can find seed oil-free options. They also have a really cool product scanner, so you can take out the app and use the camera functionality of your phone to scan products in grocery stores and see what's actually in these things. Do they contain seed oils? Do they contain other things that you want to know about out that aren't necessarily on the box. This will help you find places to eat and find foods to eat that actually have what you want in them and help you avoid those that don't. So if you're looking to cut back on your seed oil consumption, or you just want to know more about what's in the foods on the shelves at the grocery stores, download Seed Oil Scout today. If you go through the link in the episode description, click into that and you'll see a link to the Seed Oil Scout app. If you go through that link on my support page, you'll be directly helping the podcast and you'll be able to download this really cool, easy to use app that will help you find healthier food options. So go through the link in the episode description and download Seed Oil Scout today.